Interesting one, or an interesting start anyway. So my name is Kristen, biochemical oceanographer here, and I specialize, my group specializes in the study of the biogeochemistry of trace metals in seawater and the organic ligands that bind them. And so I'm going to start with, what are trace metals and why do we care? So this is a periodic table, probably very familiar to all of you, one way or the other, hopefully. This particular periodic table has been colored in or shaded in according to the concentration of these elements in seawater. And I didn't make this, and I made this joke in ChemOcean when I showed this. At some point, I use this so much, I'm going to have to remake it, because the people who did used kind of a backwards color scheme, if you ask me. So the highest concentrations are in cool colors to orient you, and the lowest concentrations are in warm colors. This is the only figure I know of that does that, so I'll try to kind of keep you on track. It's very hard to see the text in here, but each of these color blocks represents a change of 1,000-fold, so 3 orders of magnitude. So the purples are 1,000 times more abundant than the blues, et cetera, et cetera. And what you can see right away is that the most abundant elements in seawater, not surprisingly, are your salts, right? Sodium, chloride, magnesium. The macronutrients in seawater, so nitrogen, phosphorus, those are in this kind of turquoise color. They are 1,000,000-fold less abundant than your salts, so they're in micromolar concentrations. The trace elements that we study in my lab, these kind of bioactive trace metals, are these transition metals here in the middle, and they're colored in green. So they're 1,000-fold less abundant than the macronutrients. Their concentrations are low, right? So nanomolar concentrations, 10 to the minus 9 molar, in some cases picomolar concentrations, 10 to the minus 12 molar. This makes them really challenging for us to study analytically and to collect samples. We go out to sea on large metal, iron, rusty ships, right, with copper-based anti-fouling paint, and I'm trying to get samples that represent the water and not the fact that we showed up there that day. So this is has always been a challenge for us in trace metal biogeochemistry. It also means that we're doing a lot of research that we weren't capable of doing in the past. And so there's been a lot of really fun developments, and I'll highlight some of the ones that my group has gotten to participate in. But this is also a challenge for the phytoplankton in the oceans, because the bioavailability of trace metals influences the productivity of phytoplankton. So trace metals play important roles in the productivity of phytoplankton in the ocean. So they've got to figure out how to get the trace metals that they need to. This figure, or set of figures, is from Francois Morel and Neil Price's review in Science in 2003. On the left is a depiction of the nitrogen cycle, and those of you who are just getting started in chemical oceanography, you will learn this well. In this nitrogen cycle, each of these colored boxes represents enzymes that are required for that nitrogen pathway, and the element noted in them is the metal that's required as a cofactor in that enzyme. So what you're seeing here is which elements are essential to this path in the nitrogen cycle. And what you see is iron is showing up a lot. So iron is really important in nitrogen cycling. On the right here in this panel, the biomass, this is a phytoplankton cell in a box. Usually we use spheres, but they used a box. Go figure. But this is your phytoplankton. The phytoplankton's got to have nitrogen, phosphorus, and carbon in order to grow. Those, as we showed in the beginning, are relatively abundant compared to trace metals. But in order to acquire the nitrogen, phosphorus, and carbon that they need, they use enzymes that require trace metals. Again, you see iron is required in these enzymes to be able to take up nitrogen. So again, iron is really important to nitrogen uptake. Iron's not the only trace metal that matters. That will be hard for you to gather because I'm going to spend a lot of time talking about iron. But the other trace metals matter. We actually know, and to some extent, less about them. And iron, as I'll illustrate, is important globally. But zinc is really important when you're trying to access phosphorus when phosphate concentrations are low, so when you're in oligotrophic systems and organic phosphorus is more abundant than inorganic, zinc becomes really important. Zinc's also required in carbonic anhydrase, the enzyme that you get inorganic carbon uptake from. Cobalt and cadmium can be substituted. So there are lots of trace metals involved here, but iron is a big one. And in fact, insufficient iron means that phytoplankton don't grow optimally in those waters, and they can't take up the nitrogen that they need. So this plot is showing dissolved nitrate concentrations in the surface ocean. The data is from the World Ocean Atlas. High concentrations are in warm colors, okay? Everywhere forward, high concentrations are warm colors. Low concentrations are in cool. And you can see in the Southern Ocean, the equatorial Pacific, and the subarctic Pacific in particular, we have residual surface nitrate concentrations. The phytoplankton can't take up all of that nitrate. And those regions we call HNLC, high nutrient, lower than expected chlorophyll, or HNLC regions. And these regions are limited by iron. So on a global scale, iron is an important limiting nutrient in the oceans. There are large regions of the ocean where there's not enough iron. And this is important not just obviously for phytoplankton productivity, but for all the things that lead from there, including climate. One other way to look at this, and again, trying to illustrate that it's not just about iron, even though I think after this slide, that's all you're really going to hear about. On the x-axis here, this is a review Mark Moore did. Looks at the concentration that phytoplankton need, so in terms of what they have in their cells. Phytoplankton, they take up the nutrients that they need, so what's in their cell typically represents what they need, because otherwise they die and they're not there. And here's the concentration in seawater. This dashed line represents nitrogen, is normalized to nitrogen, because in much of the ocean, nitrogen is actually very, very low, right? And so when you look at this, anything that falls above this line is relatively abundant with respect to the nitrogen needs. Not something you expect to see limiting phytoplankton in seawater. If it falls below that line, it can be a limiting nutrient. You see iron is down here. Iron's not alone. Manganese and cobalt also fall below the line in this synthesis, suggesting maybe we should be looking at those elements as well. My lab studies all of these trace metals, but a lot of our research is really funded based on iron, and that's what I'm going to highlight today. All right, I keep talking about the bioavailability of trace metals being so important, and I'm not saying the concentration of trace metals is so important, and that's because unlike macronutrients generally, trace metals have very complicated speciation. They exist in different chemical forms in seawater. Those include both inorganic and organic forms. So we describe the inorganic species as iron prime or copper prime, and the organic species as iron L or copper L, where L represents these organic ligands. Organic ligands are simply organic molecules that bind to that metal. In seawater, the dissolved concentration of iron, dissolved meaning what goes through a 0.2 micron filter, so what's not sinking basically, is a sum of the inorganic and organic forms. I'm going to walk you through the examples for iron and copper because they provide kind of two contrasting images of bioavailability and speciation. For both cases, and in all cases really, the inorganic species is considered the most bioavailable. This is the form of the element that the cells have evolved to take up the most rapidly. So copper 2+, iron 3+, these are the free metal ions, free hydrated ions. And this model of bioavailability is referred to as the free ion model. So this is kind of our baseline knowledge of iron or trace metal bioavailability. The organic species we are increasingly learning are bioavailable as needed. So if the phytoplankton can't get enough of the inorganic form, they will find a way to get the organic form. Never underestimate the biology. They will find a way. For iron, organic ligands overall increase the bioavailability of iron, and that is because the inorganic solubility of iron in seawater is vanishingly low. Iron is very reactive. It reacts with oxygen. We have a lot of oxygen in the modern ocean and in the modern atmosphere, so the solubility of iron is very, very low. 99.99% of dissolved iron in seawater exists as organically complex iron, iron L. So if they can't take that up, they don't survive. So in that sense, organic ligands increase the bioavailability of iron. For copper, organic ligands decrease the bioavailability. So copper doesn't have the same solubility problem as iron. Copper 2+ is the most bioavailable form of copper. Copper 2+ is the most bioavailable form of copper. And so organic complexation means there's less of this dissolved copper in this form, which includes copper 2+, than in this one, and therefore your bioavailability is lower. That becomes really important in environments where copper is high. We contribute a lot of copper to the environment. So we have copper-based anti-fouling paints on our boats. Yeah. We have a lot of copper in brake pads in cars, so runoff from surface streets. So urban environments, coastal environments tend to be elevated in copper, and it can be toxic. Organic ligands mediate that toxicity by mediating bioavailability. When we think about the rest of the trace metals that we look at in my lab in terms of kind of similarities, cobalt is iron. It's also very reactive. It won't stay in solution if it's not organically complex, so organic complexation increases the bioavailability. It keeps it in the soluble phase where it's not lost by sinking onto particles. Zinc and nickel are more similar to copper, we think. But we know much, much less about the organic complexation, the role of ligands, and speciation for elements other than iron and copper. The vast majority of our research has been done on iron and copper. All right, so from now on, we're going to focus on iron because that is really what we're working on right now. Now, I think to highlight the role that organic ligands play, I want to highlight how they really define iron cycling. They really govern the role, how iron is cycled in the oceans. And this is a model output from Alessandro Tagliabue's lab, and it's showing the source of iron, or the input of iron, that is driving carbon export anomalies in the surface ocean. So we have different colored boxes: sediments, dust, hydrothermal, and a combination thereof, noted as multiple. All of these sources of iron are particulate. They're not dissolved sources of iron, they're particulate sources of iron. Particulate iron will, like particles do in the ocean, sink out. The phytoplankton live in the surface ocean. They need dissolved nutrients and micronutrients generally to grow. So this is where organic ligands come in. They decide how much of that particulate iron gets into the soluble phase. And I'm going to show you that through an experimental data set that came from one of the students that worked with me when I was a research scientist in Bermuda. So what this student did is we collected aerosol samples. We collected dust samples from the atmosphere in Bermuda. We then took those dust samples and we put them in seawater, and we modified that seawater in incubations with different organic ligands. So we used a couple of very strong iron-binding ligands. These are siderophores, a marine siderophore, so something we know is existing in the ocean. Siderophores are iron-binding ligands, organic ligands, produced by bacteria for the purpose of iron acquisition. So these are targeted molecules. They're very strong iron-binding ligands. They're the strongest, in fact, that we know of. And desferrioxamine B is another type of siderophore that's actually produced more in terrestrial systems, but it's commercially available and cheap, so we use it often in experiments. But these are two strong ligands, and then we used some weaker iron-binding ligands, and we looked at if we modify the water with these different types of ligands, does it matter, or do we just get the same amount of iron in solution? So the first samples we looked at this is the dissolved iron that leached from those aerosols, and what we saw is there's really not a lot of difference. So in the dissolved fraction, again, what goes to that 0.2 micron filter, we see that pretty much the same amount of iron comes off the particles, whether we use strong ligands, no additional ligands, the ambient seawater had ligands, of course, or weaker ligands. You kind of see this hint that in the longer leaches, each successive color is a longer leach, that maybe there's a little bit more coming out in these, but there's no significant difference. But we took that dissolved fraction and we filtered it again through a smaller filter so that we could look at what was in the truly soluble fraction. This is the size fraction of less than 0.02 micron, and the fraction that constitutes your soluble. This is what the cells can actually access directly. It doesn't include the little colloids, which are kind of teeny tiny particles that stay suspended. And here you see there's a very big difference. So the addition of these stronger, and we commonly refer to these as L1 type ligands, L1 number 1, I'm strongest, type ligands, really defined how much iron was solubilized from these aerosols. So L1 strong iron-binding ligands are defining how much iron is solubilized from these particles. And so Matt came up with this kind of conceptual model of dust deposition in the ocean where you have massive amounts of dust coming off of the Sahara. These are some very pretty pictures of that from NASA. That dust is coming from the continental crust. Iron is the fourth most abundant element in the continental crust. If we were to do a periodic table of the crust, iron would not be in green, it would definitely be in purple, right? And remember, in that particular weird case, purple was high. So that dust falls in the surface ocean. It's in these big particles. And the first thing that appears to happen is those particles break up into smaller and smaller particles, and eventually even into colloids, which are in the dissolved fraction. So your dissolved iron goes up briefly. But those colloids will do one of two things. Either they will be solubilized into the soluble fraction by the presence of L1 type ligands, excess L1 type ligands, meaning they're in excess of the iron and therefore available to bind new iron, or the colloids will stick together, form bigger particles, and sink back out. And so this is our kind of conceptual understanding of how iron from dust ends up in the surface ocean. And the presence of these excess L1 ligands really matters. All right, and in fact, when we recently wrote a review of iron cycling in the oceans, what we kind of we're trying to convey is that this isn't just about dust. It's also true about particulate iron coming in, we believe, from hydrothermal vent systems, advective from sediments. Excess iron-binding strong ligands, these L1 type ligands, really matter. So in my lab, we're interested in what is the distribution of these ligands in seawater. What do their depth profiles look like? What are their patterns in the ocean basins? What are the sources, sinks, and cycling processes of these ligands? And how do these ligands and their cycles in turn influence the biogeochemical cycles of metals? So I hope you're all on the same page with me that trace metals matter. Iron is a big one, right? Has big implications globally. And that organic complexation is a governing control on iron cycling. And in particular, the strongest iron-binding ligands, which we refer to as L1 type ligands, are really important. So in terms of ligand distributions, one of the big things that I've done in my kind of recent career, and this was largely in Bermuda when I first arrived here before students, and my students are now starting to take on this with me, is I participated in the GeoTraces program. And this is a global ocean survey program. There are 30 countries involved in this program. We started it almost 10 years ago, and we're just wrapping up our survey phase. These are all the cruises, research cruises, that have been conducted or will be conducted by the GeoTraces program. Yellow and black have been completed. Red are still planned and upcoming soon. Part of GeoTraces is essentially measuring the concentrations and, whenever possible, the speciation of most of the elements in the periodic table. So if you're familiar with geosex, it's like geosex, but with the whole periodic table involved. For iron-binding ligands, I measured the concentrations of those ligands on the GAO3, which was in the North Atlantic, and in GP16, which is in the Eastern Equatorial Pacific, and I'm going to show you kind of a synthesis of that data. These are contour plots of dissolved iron from those. So this is that North Atlantic section. So over here is Africa. This is the Mid-Atlantic Ridge. The black is your bathymetry. Whenever you're looking at a contour plot, you've got depth here, right? We're going east to west. This is, that's Bermuda right here. This is like Woods Hole, sort of. And the black is your bathymetry. This is the Mid-Atlantic Ridge. So what you can see here is there's very high concentrations associated with hydrothermal vents in the mid-ocean ridge. We have very high concentrations of iron associated with the margins, where there's continents that have lots of iron in particles. It's more complicated than that, but you'll learn about it in ChemOsh. Here's the East Pacific. So this is leaving from Peru, the upwelling region of Peru. Again, you see very high concentrations of iron associated with that margin. This is the East Pacific rise, and we see a very large plume of dissolved iron emanating from that. This was on the cover of Science that year. When we look at the XSL1 type ligands, the part of the ligand pool we really care about, we see something very different between the North Atlantic and the North Pacific. I kept the concentration scales the same. So you can see, again, all the little black dots, those are where we have actual samples that we analyzed. This took a lot of time for me. What we see is that excess L1 type ligands are relatively uniform through the water column in the North Atlantic and present. What we see in the Pacific is that there are essentially no excess L1 type ligands in the subsurface Pacific, with the exception of around the hydrothermal vent system and in perhaps some benthic boundary layer systems here. This difference is driving a lot of research in my lab right now. And so when I highlight some of the process work we're doing, this is where a lot of that is originating from. Interestingly, the total ligand pool across these two systems are very similar. So what it appears to be happening, or what we're hypothesizing, is that over time, as the deep ocean is circulating, the L1 type ligands present in the Atlantic are degrading to weaker ligand types. We can still detect them, we still see them in the total ligand pool, but the strongest L1 type ligands are degrading. Remember, and I will hammer this into your head, first year students, thoroughly, the deep waters of the North Atlantic are young, the deep waters of the North Pacific are old. And so this aging effect appears to be important for organic ligands. All right, we will be looking again at this on GP15. We just got these samples, and so this is an active project in my lab, and I'm hoping we'll have these samples analyzed in the next year. And we'll see what that looks like. Meridional transects are very nice because you get to actually follow water masses, which allows you to make bigger conclusions about things like aging and time. All right, so question number 2, what are we doing about, understanding sources, sinks, and cycling processes of these ligands? So we are really interested in the production of strong iron-binding ligands. One of the sources we've seen for L1 type ligands is associated with phytoplankton communities. So here, this is work for my postdoc. Again, we're going back and repeating some of this in more detail. All you really need to know here is we incubated phytoplankton in the Southern Ocean and in the California Current, which is another iron-limited system. When we grew up iron-limited diatom communities, we saw big increases in excess L1 ligands. And so we've hypothesized this is related to diatom and bacteria associations, because bacteria can produce cytenophores and diatoms cannot, but they tend to need each other for other things. And we've got a project in the Southern Ocean doing this. My PhD student, Shannon Burns, this is part of her dissertation, is looking at how trace metals are being influenced during diatom blooms and the production of ligands. We have also been working on understanding what some of these ligands are, and one of them resulted from conversations between Maya Breitbart here at CMS and myself, and we started talking about we wanted to find a way to work together because we're good friends, and so we started talking about our different questions that we had in our field, and it came through that conversation that it turns out viruses, in terms of size, are in the dissolved fraction. So they fit through that 0.2 micron filter that I measured dissolved iron and iron-binding ligands in. Viruses that infect E. coli, so in model systems distinct from the oceans, have iron in their tails. So this is what a virus looks like. They're very freaky looking. These are what they mean by tails, and they have little iron atoms in the tails. And so Maya and I got together and we wrote a hypothesis paper hypothesizing that if viruses in the ocean also use iron in their tails, then viruses would comprise a form of dissolved iron in seawater. Very different than the way I had thought about this chemically, because I didn't think of biology in the 0.2 micron size fraction. That's what that filter is supposed to do, is get rid of that stuff. But it turns out viruses, get into everything. But viruses are also organic. So this means that viruses not only would be a part of the dissolved iron pool, they'd be part of the dissolved iron binding ligand pool. And so this is something that we are still doing some research on and are very excited about. And when we look at ligands, this is kind of what we know about them. So a lot of vagueness. We have a lot of ideas about what ligands are, but we don't have a lot of structural information, although there's some really great mass spec developments out there that our collaborators are using to get at that. But viruses are now included in our possibilities. All right, so in terms of cycling of ligands, we have two active projects looking at time series. So I talked about how we see this effect between the basins. We're also very interested in the temporal changes in the iron speciation in the upper water column. And so we're doing that first at the Bermuda Atlantic Time Series study. So BATS is a long-running time series program in the North Atlantic. It's been running since 1988. We've been collecting samples over the last year, and we'll continue for another 2 of those cruises, and we'll be measuring the speciation of iron in the upper water column. Salvo Caprera in my lab, a research associate, will be doing the iron speciation analyses for that. And he will also be looking at the evolution and changes in iron speciation in sea ice. And so we're part of the Mosaic project, and this project is crazy. So they are taking a German ice, they are freezing it in the Arctic ice. They actually leave next week. So they're allowing the ship to be frozen in the ice, and they're going to drift with the ice for 350 days. They will set up this very comprehensive sampling setup with different tents and communities and this whole thing. And we will be getting ice cores every month for almost a year. And we will be able to look at the iron speciation in those ice cores to see what happens when the ice forms, how the speciation evolves within the ice and then during the melt. And this is important because the sea ice is also hypothesized to be an important source of iron and ligands to the surface ocean in the Arctic. And they're modeling this expedition. They're trying to recreate, for those of you who know about it, the Nansen Fram expedition, back in the day. So it's a very cool project, and they have a lot of neat stuff online if you want to check it out. We're having fun being involved in that, and we're also excited we're not spending 350 days on the ship ourselves. They're not. They're flying people in. They have a whole thing. They're building a runway on the ice, and it's a whole thing. All right, so the third aspect of the research in my lab is how do ligands and their cycles influence the biogeochemical cycling of metals? So we've got a couple of big collaborative projects for this. We're working with the exports program, which is, I feel like it's like movies. We're kind of recreating a lot of the stuff we've done before with some new CGI. So exports is like a recreation of JGOFs. So this idea of looking at carbon export in the surface ocean. And this time, we have extensive satellite data going into this. We did our first cruise last year, and it was on the Ravel and the Sally Ride. So it was 2 ocean-class vessels full of scientists. I mean, these are big operations in oceanography that we've got going on. And we have autonomous vehicles. It's a whole thing. And so my lab is participating in this, looking at the role of iron. As we are all in agreement, iron is really important. Yeah, right? And diatoms are a phytoplankton that is really important to carbon export. And the relationship between how iron is driving diatom growth and diatom community structure is a really important question in understanding carbon export in the surface ocean. And so this is our role in the exports program. We have our second cruise for that coming up in the Atlantic early next year, and that will be on the Atlantis and the Armstrong. So another dual ship effort, and Salvo Caprera and my student Shannon Burns will both be on that. And this will hopefully be the last chapter in Shannon's dissertation, although as my students will testify, I tend to keep adding, so we'll see. We also have a project that was just recently funded to start looking at the effects of ocean acidification on iron speciation. So this is a map of pH in the oceans from Taro Takahashi. We'll be doing our field work up here in the subarctic Pacific, where as you can see, pH is especially low. And the idea here is we're going to be modifying the methods we use to measure iron complexation or iron binding ligands at different pHs, and then conducting a series of field studies, experimental studies and manipulation experiments to look at how speciation is changing as a function of pH, but also how that influences the bioavailability of iron, which of course is a really important question as we continue changing the ocean system. We have a lot going on in my lab right now, just in case, whoa. So we also have been collaborating with Maeve Lohan and Alessandra Tagliabue out of the UK to really look back at hydrothermal vent systems. So I showed you that picture of that data from geotraces of the XSL1, that plume of XSL1 apparent production associated with the hydrothermal vent system was really quite surprising to me. And then this came out of Brandy Toner's lab, where they looked at gene expression, and they found bacteria expressing the genes for siderophore production in hydrothermal vent plumes. So that suggests that we have a siderophore source associated with hydrothermal vents, which is really interesting, and that would be an L1 type ligand. So we are very interested in trying to look at that. So Travis Mella, a PhD student in my lab, is wrapping up his dissertation with a really cool chapter looking at the cycling of iron in incubation experiments that we conducted in near field and far field hydrothermal plumes from Rainbow and TAG, as well as one from Lucky Strike. So we have a really nice data set coming together of the trace metal cycling and the ligand characteristics in the soluble and dissolved fraction. So stay tuned, because he'll be hopefully showing that in his defense here in a couple months. And then finally, as Bob alluded to in his talk, we've recently become involved in looking at the role of iron and other nutrients in the harmful algal bloom dynamics on the West Florida shelf. So we've gotten some funding from the state of Florida, and we'll be doing some field work as well as some laboratory work to look at the role of iron in the HAB initiation. And so this is something that is really new to me and really, really interesting, but at least one of the hypotheses is that dust deposition from the Sahara, which lands on our ocean here on the West Florida shelf in the summer, is driving trichodesmium blooms, and trichodesmium is a cyanobacteria that's also a diazotroph, so it doesn't need nitrate in the water. It can use nitrogen gas out of the atmosphere. That trichodesmium then is using nitrogen out of the atmosphere, creating organic matter, and it therefore is releasing fixed nitrogen into the surface waters, which may be part of the fuel for Karenia brevis. And so we'll be trying to test those hypotheses in field studies and lab work with fish and wildlife. We're also looking at the effects of the proposed clay mitigation strategies on trace metal chemistry. The clays are, aluminum-based clays, and so they want to know, are we going to have some unintended or unexpected outputs of that in terms of metals. So we'll be doing that, and you'll see some of that data coming up soon as well. So this is the kind of work we have going on in my lab right now. Let me know if you have any questions.